Today, we will discuss in detail regarding other cGMP guidelines. cGMP means current GMP guidelines, and we will also discuss the process flow of various formulations in the industry. Coming to the cGMP concepts, so what are GMP? GMP is nothing but good manufacturing practices. So yesterday I have told you we have discussed an example of the cGMP guideline with example of US FDA. Similarly, the points will be same for all the countries, but the naming and nomenclature will be different. See, the cGMP concepts, what are good manufacturing practices? So good manufacturing practice for medicinal products, quality assurance. In quality assurance, you should follow GMP. In quality management, you should follow GMP. While documenting anything, you should follow GMP. In holding and distribution of the drugs, you should follow GMP. In materials, any activity related to materials like procuring, dispensing, manufacturing, packing, everything, you should follow good manufacturing practices. During the equipment operation, you should follow good manufacturing practices. While construction, you should follow the good manufacturing practices, and also while working in the premises, you should follow the good manufacturing practices. Personal training and personal hygiene is also a good manufacturing process practice. You should follow good, and you should have proper hygiene. And regular quality inspections or audits like self inspections, audits, supplier audits, and their approvals should be done as per GMP. And all the contract production and the analysis of the material should be done as per GMP. And complaints and product recalls documentation should be done as per GMP. Qualification and validations, sanitation and hygiene, quality control activities, in all these sections GMP is applicable. So based on the guidelines framed by the various regulatory bodies like USFDA, TGA, MHRA, CDSCO, and other country regulatory bodies, there is an institute in India called GMP Institute. This GMP Institute has all, what it will do, it ensures that all the companies will follow the good manufacturing practices. It has taken all the guidelines into consideration, and it has framed, there are 10 principles are framed by the cGMP Institute in India. So we will discuss each principle one by one. The first principle is writing detailed step by step procedures that provide a roadmap for controlled and consistent performance. So what it is telling that we should have a proper standard operating procedures that are designed so as to understand, even a person, fresher person who is entering an industry or the manufacturing area, from the SOP he should understand how to run the mission, how to operate, how to clean, and how to walk, how to wear the proper gowning, how to wear the protective equipments, everything should be done. So the first principle is there should be a written procedure or SOPs for each and every activity that is being carried out. Second, and what is the importance of the written procedures? The procedures or the SOP should be very clear, concise, and logical. It should be concise in brief, but it should be very clear. And when you are framing an SOP or writing a written procedure, you should ensure that you should never deviate a written procedure without the approval of a supervisor of the quality department. So it is telling that you should follow according to the working procedure or written procedure or the SOP. So this is regarding the first principle. And second principle also is an elaboration of the principle one, where it is, principle one is telling that there should be an SOP. Principle two will tell that you should follow that SOP. Why you should follow the SOP? Due to, because if you follow the SOP, you will do your activities in a controlled manner so that there will be no contamination of drugs, there will be no mix ups, and there will be no errors. This is the second principle. Principle 3 is talking about you should document all the work promptly and accurately. We will discuss regarding this in section ALCOA Principles in this session only. So what is principle 3? Principle 3 is telling that you should document every work what you are doing. And in pharma industry, the main thing is do what you write and write what you do. These both are interrelated. And principle 4 is proving that the systems do what they are designed to do by validating. It is talking about all the equipments on the mission should be validated. This validation concept will be very deeply discussed with you all in two sessions. One is the process validation session and another will be cleaning validation section. In the forthcoming sessions we will discuss this. So the summary of this is: validate and document the work to prove that equipment is working consistently what it is supposed to do. So it should provide a quality product. What it is the equipment is supposed for, it should provide a quality output or the quality product. So the equipment should be validated and qualified. All this validation and qualification should be documented. This is the third and fourth principle. Next principle is the design of the facility and the design of the equipment from the beginning. It should be such that yesterday also we have discussed the same things. So the facility should be designed in a proper way such that the movement should be uniform. There should be equipment shall be established for easy operation and cleaning and movement of materials to without any cross contamination and avoiding the mix-ups. And also, once you construct a facility and install the equipments, the work is not completed, right? So it should be properly maintained at regular intervals with proper cleanings. This is the concept of principle 5 and principle 6. So the area and facility should be such that there should not, we should remove any unnecessary traffic in the production area. All the material should be segregated batch wise, and their components also should be segregated to minimize the mix-ups and errors and contamination. So, and also the facility design should be such that HVAC unit will function properly with maintaining proper temperature and RH in the manufacturing and packing areas. This is the principle 5 and principle 6. Next, principle 7 is clearly defining, developing, and demonstrating the job competence. So once you are joining in a company, you will sign a document called job description or JD, which will tell what are all the works you are trained in and what are the activities you should be involved. Any activity in which you are which is not mentioned in your job description, you should not perform in the company. So the job description should be clearly defined. This is the principle 7. This is the main part of GMP. So uh, what will job description have? It will have all the activities in which you are trained in. So the training is an important step for the GMP and all the equipments and manufacturing and cleaning operations in which the person involved in the activity should be trained upon. So the main concept is doing right first time. The first time when you are doing the activity, you should do it correct. There will be no chances, there will be no excuses, because one mistake in a production department, the entire batch is lost and so much financial problem to the company. Next, principle 8 is protecting the products against contamination by making and maintaining cleanliness as a continual habit. So when you are manufacturing and packing a product, you should be very careful that you should be protected from the drug and also the drug should be protected from you. So how you will protect the drug and yourself? By wearing a protective clean clothing. Every day when we are going to the office, everyone should go in a clean manner, but after taking bath, and there should be no very clean shaves, there should be no nails, there should be no hair fall, and if you are sick or ill, you should not go to the company. All the things should be followed. All these things, how you are wearing your secondary gowning in the manufacturing area, processing area, also should be followed as per the SOP. And inside the manufacturing and packing areas or laboratories or storage areas, you should not smoke, you not eat, and not drink. Also drink water also, it should be done only outside the areas. And inside the areas you should not keep any plants. And even the tables and chairs which you are using inside the process area should be made up of SS316. Wooden chairs etc should not be used. Why? Because it will cause contamination and it is difficult to clean. Only stainless steel 316 grade is easy to clean to avoid the cross contamination, right? This is principle 8. Principle 9 talks about how we will build quality into products, how you will produce a quality product. You should control all the raw material, packing material, everything. You should control the manufacturing process, you should control the labeling and packing process, and you should have control on holding and distribution also. If you are having control on all these steps in the pharma industry, then ultimately the output or the final product will be of high quality. Principle 10 is how you will maintain now high quality. You cannot believe anyone, right? So yeah, every time the quality assurance department should conduct self inspections and periodic audits should be done for the other departments to ensure that they are functioning as per the SOP or written procedures. So till now these are the 10 principles that is framed by the GMP Institute that are to be followed in India. And next, we will discuss on another concept. It is called ALCOA plus principles. ALCOA plus principles are being followed in the pharma industry in all the companies to maintain the cGMP activities. This is very important in GDP. Till now, we have discussed regarding GMP. Now, what is GDP? GDP, good documentation practices. Good documentation practice, the first and foremost important thing you should know when you are documenting anything related to a product or a batch is you should follow the ALCOA principles. So, what is ALCOA? A means attributable, L is legible, C is contemporaneous, O is original, A means accurate. Then what is plus? This plus is recently before this has come into picture only 5 6 years back. Previously it is only Alcoa. So now what is meant by plus? Plus is nothing but anything additional, like it should be available, it should be enduring, consistent and complete. So all together all these things, yeah, the 9 things they are called as ALCOA principles that are to be followed in a pharma industry. So first, what is attributable? Attributable is nothing but uh, for suppose someone, I am writing a document, so I should sign it. Whoever does the activity should sign it with his sign. On which date he has performed the activity, he should mention the date also. This is called attributable. And legible, L means legible. Legibility means, you know, no, legibility means it should be clearly readable once after it is recorded. Next is contemporaneous. Contemporaneous means online documentation is called as contemporaneous. For suppose you have done the drying activity today, you should complete the drying activity online today only. You should complete. Once you start the drying, you should mention the time in the batch manufacturing recorded as at 8 o'clock or 8 to the drying has been started. If it ending has been completed at 8:10, at 8:10 drying has been completed. So timing should be very contemporaneously or onlinely it should be recorded in the batch records. Next point is O. O comes for original. Original means there should not be any Xerox papers in the batch record. Everything should be only original documents. It should be in an unaltered stage. And next is accurate. Last A is accurate. Accurate is nothing but it should, the data reflects its actual value, free from error or trueness. So there is no data integrity. Accurate means correct information you need to provide in the batch record. Next, we will talk about the plus principles. The first one is available. So until the product is expired, the batch document should be available for review by anyone at any time. Next is enduring. Enduring means you should ensure that all the records are existing in the plant and available for review, or if any auditor asks, until its entire period it should be available. Means entire period in the sense it is the expiry of the product. Next, complete. All the data should be complete without any missing pages. Entire batch record along with the supporting printouts and documents should be made available. This is called complete document. And next, consistent. Consistent means data should be done in a sequential manner with sign and date. Every person who is performing the activity, he should sign it, and date should be also done when he has done, and follow the GDP, good documentation practices, for consistency during documentation. This is called as consistent. So this is the minimum requirements that are to be followed while writing a document in a pharma industry. These are altogether called ALCOA principles. Next, we will jump into uh, this section describes type of the pharma plants. This section describes various type of pharma industries existing in India as well as all over the globe. Various type of pharma industries means we will get an overview of the different type of formulations and their brief flow chart. So actually there are two types of industries. First is the API industry and second is the formulation industry. Formulation industry, again it is divided into oral dosage forms. Second is steriles. Afterwards it is biologics, semi-solids, parenterals, everything comes under formulation industry. Then we will go into each type one by one. First is oral solid dosage forms in a formulation. First is oral solid dosage forms. Next is liquid orals, steriles, semi-solids, and biologics. So now we will go and discuss the flow chart or the flow pattern in each type of industry, how the manufacturing. Just we will discuss an overview just for the understanding. First is the API manufacturing process. In API manufacturing process, the various processes that are included are sometimes synthesis, distillation, extraction, crystallization, purification, filtration, drying, milling. These are the various processes that are to be followed or used during the manufacturing of APIs. See how it will be done. First, raw materials will be taken. They will be kept in a reactor or distillation and provide sufficient amount of temperatures for them for the and allow for the reactions to occur. Next, anything crystallization will be done. This crystallization, all the crystallized solids, they will separate it from the liquid or the other things. And next, this separated crystals or the solids, they will be dried. And once it is dried, the API intermediates are of the final APIs. And once after processing of the final product, then the API, it will be packed in a required form. Like if the customer requires it in a crystal form or milled powder or in a liquid or the type of formulation of the API, we will pack the product and send it to the customer. This is regarding the brief of API manufacturing process. Next, oral solid dosage forms. We will take an example of a tablet, how it is manufactured. I will tell you the flow chart. So first, we will receive the raw material from the warehouse. Next, verification of the materials and wait at the shop floor. Shop floor means the manufacturing area. Verification, who will do production, and QA person will be involved in the material verification. Why? Because now the material is transferred from warehouse to manufacturing area. You should check that accurate amount which is dispensed or weighed in the warehouse should be brought into the manufacturing area as per the batch document. It is done. Now after verification, what you will do, you will take the line clearance of the manufacturing area by the QA, and then you will proceed for manufacturing process. The first step is you should sift all the materials and then go for dry mixing and wet granulation. After wet granulation, you should remove the water which is added, right? So next, this step is called drying. This drying will be done in a fluid bed dryer or fluid bed processors. And after the drying, you should ensure that the loss on drying or the moisture content should be within the limit. For that, you should test it within the shop floor. And next, after drying, now the granule should be of uniform size, right? So you should do it, you should mill the granules or size the granules by using a multi mill or combi mill or a cone mill. Like that, there are different types of mills. You should use the mill which is validated for that particular product. Now once the milling has been done, it will be carried out to from granulation area. Now the material will move to the blending area. So again, that area line clearance should be taken from the QA, and then lubrication should be done. Now again, QA role here is you should send a sample for analysis. This is called sometimes content uniformity or the blend uniformity samples will be sent, and also sometimes bulk analysis will be sent. Next, you should calculate the yield. Yesterday I have told you there are three types of yield. First is the actual yield, next is the theoretical yield, next is the percentage theoretical yield. At every step you should calculate the yield. If any deviation from the specified limits, you should log a QMS document like incident or deviation. Next, now you have got the report of the bulk analysis. Now it is approved. Now this blend should be taken into the compression area, and you should take the line clearance by the production and the QA. And next, you should pre-compress it by setting all the required parameters of tablet weight, hardness, dt, friability, everything, group weight, everything. Now if all are okay, you should take the startup clearance from the QA person and perform the compression activity. And once after the compression activity has been done, you should do the visual inspection. Visual inspection, or it is called as AQL, acceptable quality limits. Acceptable quality limits, you will have a checklist in which there will be the different types of defects are classified, like critical, major and minor defects, and based upon these defects you should approve the compressor tablets for the next step. So what is the next step after compression? If everything is okay and all are within the limits, if the AQL is within the limits, AQL is a separate guideline which is framed by the ISO, Indian Standard Organization, wherein you have to collect the random samples from all the compressor tablets, or any stage you should collect the random samples and do the AQL check. I will show you a how the AQL sheet will look like in the next sessions, a sample thing. What are the types of defects also I will explain there. So uh, again, after compression you should calculate the yield, and next you should take the tablets for the next step, that is coating. In the coating, again you should take the line clearance from production and QA, and next you should carry out the coating process, and then again you should do visual inspection. Visual inspection means again it is AQL, and after AQL is passed, then you should calculate again the yield. So now the coated tablets are completed. Now they are ready for packing. Now you should, the material will be again taken to the packaging area. You should again packing material will be dispensed from the warehouse. This packing material will be verified, and then necessary batch coding. Batch coding means printing of the batch number, manufacturing date, expiry date on the packing material. This should be done after taking the line clearance of the IPQA, and next, and the packing activity will be done. And terminal inspection. Terminal inspection means at the end or on, and also online of the packing activity also you should carry out the inspection. So once after the packing is done, the IPQA person will collect the samples for analysis, reserve samples, control samples, stability samples, and all microbiology samples. It should be collected and sent to the QC. And by the time this packed products will be kept in the hold area or warehouse. And once the result is come, the finished goods analysis has been done and the result has come. Now it will be transferred to the FG store. FG stores means finished goods store, and BPR or the batch packing record will be sent to QA. And next steps will take place, that is the batch release, everything will be done from the QA. I will discuss with you regarding the batch release process in a separate session. This is regarding oral solid dosage forms flowchart. Next is the liquid orals. How a liquid orals will be manufactured. See, first receipt of the raw materials, sampling, and after the raw materials are sampled in warehouse by the QC, analysis will be done, and all the approved materials will be then dispensed according to the batch manufacturing record. Now they will take to the manufacturing area. In the manufacturing area, as like oral solid dosage forms, again checking of the dispensed materials will be done, line clearance will be taken, you and manufacturing will be done. The mixing will be done for liquid orals in big vessels. It is entirely liquid. No, the API will be mixed along with other solvents like sucrose, sugar solution, or based upon the taste enhancers, all will be mixed in a water base as per the quantities mentioned in the batch manufacturing record. Now it is manufacturing has been completed, and the product should be stored somewhere. It will be stored only in the manufacturing vessels, and the IPQA will collect the samples and send it to the QC for analysis. If for liquid orals, what is the main thing IPQC test? It is the pH, viscosity, test should be SA. These three are the main things that are to be conducted for the liquid orals. Once if all the tests are passed in QC, you will get the COA, certificate of analysis, which is approved by the QA. And then only you should proceed for packing. For packing, what is required? Packing material is required. What are the packing materials for liquid orals? It is a bottles, right? So again, packing material will be received, sampling will be done, testing. Only the approved packing materials will be issued for the manufacturing and the packing activity. Now these packing materials will be taken to the manufacturing area, and the bottles will be washed here. Next, the liquid will be filled, and again sealing will be done. And after sealing has been done, visually you should inspect the IPQC checks at various intervals defined in the batch manufacturing record for the seal integrity, so that there is no leakage of the liquid from the bottles. Once this is done, you should go for the labeling and the packing operations. Labeling operations, again you should take care how that the correct batch number, manufacturing and expiry date has been printed on the labels, and then you should go for final packing. The sample will be sent again to QC, and we will wait for the analysis. The material will be sent to the finished goods warehouse, and after release it will go for the QA release. It will go to dispatch. This is regarding liquid orals. The next type of formulation is parenterals. Parenterals are very critical. They are called as a parenterals or steriles. This should be taken, and manufacturing should be done very carefully only by authorized responsible minimum number of persons. More number of persons should not enter the parenterals manufacturing area. The entry should be restricted. Why? Because if the more time number of manpower is entered into the production area of the parenterals, there will be more chances of high contamination. So number of persons working in sterile areas will be very less. uh, so the process, the main thing in parenteral second one is, all the water that is being used in the manufacturing process, it should be sterile, or it is called as the water for injection. The water should be tested in QC for sterility. There should be no microorganisms. And also, while manufacturing the parenterals, the conditions of the room are also very strict, such that clean room conditions, there are types of rooms like class 100, class 1000, class 10,000, 1 lakh rooms are there. These types of classes of the rooms, it is given by the ISO guidelines. For steriles, you should follow very, like class 100 or 1000 you should follow for steriles. And for orals and liquids, everything you can follow more than class 10,000 also, it is not an issue. So in brief, the process flowchart for the parenterals is: first, this is the warehouse area. From the warehouse area, the API will be taken into the manufacturing area. This is called the preparation area. The preparation area, in the preparation area, in the preparation area, first the store materials will be taken to a pre-cleaning area. Here dedusting will be done. From here it will go to the preparation area, where the API will be mixed with the water for injection in dedicated tested vessels. Mm, uh, After satisfactory line clearance, once it is mixed, now this injection solution will go to the filling area. This filling area, again, it should be sterilized, and also it should be cleaned. And regular sterilization of the areas should be done. So in this area, filling will be done into the vials or ampules. Once this is completed, uh, before and after manufacturing also, the area should be made sterile, so that there is no microorganism. Because if there is a microorganism in the room, the microorganism will also enter into the vials or ampules of the product, and it will be completely contaminated, and it is unfit for use. Now after filling area, now it will come to the quarantine area, where it will be stored until the result of the vials is obtained. Once after satisfactory results, or once when it is all the results are within the specifications, next secondary and tertiary packings of the vial will be done, and then sent to the finished store warehouse, and then finally sent for shipping after QA approval. This is regarding parenterals. Next, another type of formulation is called biologics. What are biologics? Biologics are nothing but they are the uh, type. For example, insulin injections, it is called as a biologics. Paracetamol injection, it is called parenterals. Or ibuprofen injection is called parenterals. Biologics is nothing but any sterile dosage form that is manufactured from a living organism is called as a biologics. This is a completely different and novel type of formulations. Okay, how it will be done? First step is inoculum. Inoculum is nothing but the cell or virus. It will be grown or matured from a small single virus to more virus. It will be matured under specific conditions. Next, this one, it will be taken to the next step. It is called as a bioreactor train, where the cells will multiply, and until the required quantity is obtained, you need to provide the sufficient temperature and RH and other feed for the cells to multiply in reactors. And once we have achieved it up to the production level, the sufficient quantity, we need to harvest those. What is meant by harvesting? Harvesting is nothing but the required amount of virus. You need to take it from the production containers or vessels, and you should filter it or separate the required thing from the unwanted cell materials. This is called as a harvesting of the virus. Next step is, we need to treat the virus, right? So what it should be done, a virus that is causing some effect to us, it should be inactivated, and only it should serve the purpose for which it is being taken as a medicine. So here you need to do pre-virus operation and the post-virus operation. In pre-virus operation, the first thing is, main virus should be inactivated. Once it is inactivated, it will be filtered by the cations and the anion exchanges, and then we will get the required virus. And then this one will be taken into an again filtration tanks, like ultra filtration and diameter filtration. Something will be done, means these are the various steps that will be done. And once it is done, final filtration in a sterile environment will be done, and then it will be taken to the manufacturing and packing area, similar to the parenterals. For you should manufacturing and mix it, and pack it in vials or ampules by using the flowchart of parenterals itself. This is the brief regarding biologics. This is a very vast subject which require very careful while working in biologics department. Next is the semi-solid formulation. Semi-solid means the ointments, everything, creams will come under semi-solids. So here, as you know, maybe you have studied in your B.Pharm or something. Here, water soluble ingredients should be heated separately. Oil soluble ingredients should be heated separately. Both should the water soluble ingredients should be kept, should be mixed with the oil soluble ingredients in a separate vessel by continuous mixing, so that emulsion will be formed. So the two types of phases will be mixed uniformly, and effect of the medicine will be shown in a perfect manner. And now, once after it is mixed, you should transfer the finished product by the pump into a drum or a tank, and from this tank, it should be filled into the ointment tubes, ointment bottles, everything. This is the brief thing regarding semi-solids. So now this session is completed as of now. So if you are having any questions, the screen is for you.